The overall goal of the following procedure is to determine the sub-mitochondrial localization of a normally nuclear cell cycle kinase, and then to analyze how its mitochondrial localization affects the progression of the cell cycle.
This method can help answer key questions in the mitochondrial research field, such as how nucleus communicates with the mitochondria during the cell cycle.
By tagging proteins with the mitochondrial leading sequence, we can take advantage of specifically overexpression of this protein in the mitochondria, so we can study their mitochondria-specific functions.
Though this method can provide insight into mitochondrial targeting of certain proteins, it can also be applied to the other organelles, such as the nucleus, Golgi, and lysosome.
After culturing, homogenizing, and pelleting cells according to the text protocol, transfer the supernatant into a new tube and centrifuge the sample at 7,000 Gs and 4 degrees Celsius for 10 minutes. Then use 200 microliters of ice-cold IBC buffer to resuspend the pellet and divide the homogenate into 2 aliquots. Centrifuge the samples again at 7,000 Gs and 4 degrees Celsius for 10 minutes and repeat the wash. After discarding the supernatant, add 30 microliters of cell lysis buffer to one of the pellets and store the lysate at -80 degrees Celsius for immunoblotting. To carry out sodium carbonate extraction,